Alrighty, so now we're going to move on to talking about cell respiration. So how we go through and recharge ATP. So this looks really confusing at first. So I just want to refresh a few things. So remember we talked about catabolism versus anabolism. So catabolism is when we break down things into smaller components. And we're going to talk about it related to food pretty much. So you have your carbs, your fats, and your proteins. And we're going to break them down into smaller subunits that we're then going to feed into some anabolic reactions. So anabolic reactions are where we're building things. We're going to be changing them into things that we need them. So for the sugars examples, we're going to use them either make glycogen or starch, or we're going to put it in the process to make ATP. For fats, you can see we can feed it into some of these other cycles, but we can also use them to make phospholipids, which are super essential. Your proteins, we can use them to make other parts for amino acid synthesis. But hopefully you can appreciate that metabolism is actually quite complex. It's kind of interesting. One of the things we've done discussion before is talking about the different type of diets and how that could affect it. If you talk the keto diet, what they want to do is totally eliminate this part of the chain. So you kind of think about, okay, how does that influence everything else? So it's kind of interesting, and hopefully after today you'll appreciate metabolism a little bit more. So just a reminder what we're doing. Our goal is to recharge ATP. So just refreshing what it is adenosine triphosphate. So we have that sugar with our adenine. I could call it wrong on the first one. And then we have that triphosphate. So we have those 3 phosphates. If you remember, the energy is these phosphate bonds. This is where the magic's going to happen. So when we need to do a reaction like break a hydrogen bond or remove a functional group, we have to pay for it. You know, just like if you want to get something from the store, it's going to cost you. Well, how we're going to pay for it is in these phosphate bonds. So if I want to break this hydrogen bond right here, I'm going to break this off right here, get a little packet of energy, and now I'm going to use that to drive the next reaction. And then that makes it ADP, and then we can get down to AMP. Now, you're going to see there's 2 terms. There's going to be aerobic versus anaerobic. So the first thing we're going to talk about is aerobic, and that's if it uses oxygen. So think about aerobics and people working out. You're going to be breathing a lot, consuming a lot of oxygen. Now, most of this is going to happen in the mitochondria. So remember we talked about it, analogy is the powerhouse of the cell. Well, the reason it gets that name, it's where we recharge ATP. Now, it's going to occur in several steps. Don't get too bogged down by the details. We'll talk about the big picture on each of them. And there's the overall equation. I think it does show up on a test question here or there. What is the equation of photosynthesis, cell respiration? This is it. So we're going to take glucose, which is this chemical structure right here, take an input of oxygen, and we're going to get a product of carbon dioxide and water. So it looks kind of scary when you first look at it, but don't freak out. We're going to break it up in 3 stages. And this is kind of like an overview slide, so it's a good summary then. So the first one's going to be called glycolysis. Basically, what's going to happen in glycolysis, we're going to take that 6-carbon glucose molecule, and we're going to break it down into 2 3-carbon molecules called pyruvate. And it's a series of 10 steps. We're not going to go in extreme detail through it. Basically, you're taking glucose, which is a 6-carbon, and converting it into 2 3-carbons. So we're going to still have 6 total, but it's like if we just chop a molecule in half. Now we are going to get a little bit of ATP off this, only 2. You know, it's something, but it's not the big payoff that we want at the end. Now what's unique about glycolysis is this is going to happen out in the cytosol, all right? So outside the mitochondria. And this is the only process of cell respiration that is anaerobic. So that means it doesn't require oxygen. And we'll hit on this when we talk about fermentation. Now what's going to happen next is you're going to take this pyruvate that we made, and we're going to feed them into what's called the TCA, or the citric acid cycle, or the Krebs cycle. Just like it says in the name, this is a cycle where we're going to take these carbon molecules and we're going to feed them into the cycle. And what we're going to do is we're going to be harvesting off the carbons. And as we start to do that, we're also going to be harvesting electrons throughout both processes. Now, these electrons are going to be driven to the very last process, then we're going to use that to regenerate ATP. Now, this part occurs in the mitochondria, or in the very matrix, in the very center of it. Now, as I mentioned, we're going to be picking up a lot of electrons throughout here. It's one way we're going to transfer some energy. And we're going to take those electrons and we're going to drop them off in what's called the electron transport chain. Sorry, or called OXPHOS for short. And this is going to be on that inner membrane of the mitochondria. So what's going to happen is we're going to take these electrons and it's going to drop them off. And what they're going to do is drive hydrogen ions and create a gradient. And then that gradient is then going to power this molecule called ATP synthase. And that is where we're going to recharge the ATP. So don't freak out. We'll go through each step. Don't worry. But let's just remind ourselves about the anatomy of the mitochondria. So remember, it's got a double matrix, our double membrane system. The matrix is the inside part, has an inner membrane and an outer membrane. Just refresh ourselves on the anatomy and how we're going to move these electrons around. So I kind of mentioned this on the previous lecture, but we're going to use electron carriers, and the big one that's going to do most of the work for us is going to be NAD+. Now, I kind of think of them as like a taxi cab, or Uber or Lyft, if you want to think of it that way. So the main job is to pick up electrons and transfer them from one place to another. And then once they get to the final location, they drop off their electrons, and now they're empty, and they go back and pick them up over again. So they just shuttle back and forth. Now, when it's without electrons, it's just NAD+, and when it gains the electrons via hydrogen, it becomes NADH. Now, another one that we'll see is FADH, FADH2. Think of one as Uber, one as Lyft. They still do the same thing. So let's go through the stages in a little bit more detail, but don't freak out. Like I said right here, relax. Focus on the big picture, like what's going in and how does it relate to the overall equation. What are the products? What are the substrate? What's getting in? What's getting out? I'm not going to expect you to remember all the stages in order, all this stuff. This is just torture. You're not a major. I'm not going to do that. What you should know is products from one stage will be used in another. So your cell's very efficient. So the big first one was glycolysis. So this happens out in the cytoplasm. This is where we took that molecule, glucose. Remember, that is a 6-carbon molecule. What we're going to do is break this into 2 3-carbon molecules called pyruvate. So in order to do that, we have to use some ATP, and we're going to pick up some electrons. Now, in the process, we get pyruvate. We get some ATP off of it, but we had to spend some, so we kind of get 2 net ultimately, and we get some full taxicabs. Now, glycolysis is broken down into 2 stages. The first half is the prep stage. So basically in the first half is pretty much through the process of chopping this in half and making it into this molecule, glyceraldehyde 3-phosphate. It's a series of like 5 steps, and it takes an input of energy to get everything prepared. In the second half, we're going to harvest the energy that we put in the first stage and convert this glyceraldehyde 3-phosphate into pyruvate. Now remember, this is the part that doesn't require oxygen. But the big takeaway that you should get from it is we go from 1 6-carbon molecule to 2 3-carbon molecules. Boom, boom. We did get a little bit of energy. Locations in cytoplasm did not require oxygen at this point. So now the next stage. So we need to prep the pyruvate a little bit. So this is where we're going to take that pyruvate and we're going to go from the cytosol and we're going to go into the mitochondrial matrix, so all the way in the innermost part. So the first thing is we need to prep the pyruvate. So how we're going to do that is we're going to take the pyruvate, boom, 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 Here's your 3 carbons, they're shown in gray. And we're going to add it to a molecule called acetyl-CoA right here. Now, acetyl-CoA is kind of like an escort, if you want to think of it that way. We want to get into the citric acid cycle, but to do that, we need a little bit of help. We can't do it on our own. So how we're going to do that is you have to be acetyl-CoA to get in. Now, what we're going to do is we're going to take pyruvate, add it to an enzyme called coenzyme A. In the process of doing that, we're going to chop off one of our carbons. It's going to be released as carbon dioxide, and we're going to pick up some electrons. So I'm going to walk you through as best I can right in this little diagram. So here's your pyruvate. There's 1, 2, 3 carbons. It's going to come into the matrix, and what's going to do, it's going to bind with coenzyme A, and in the process of doing that, you're going to chop off one of these carbons right there. And this is the product that you get. In the process of chopping off that carbon, you have now 2 coenzyme A. Now this molecule is ready to go into the next part. So the TCA Krebs citric acid cycle, it's all called the same thing, is just like it said, a cycle. So what's going to happen is acetyl-CoA, the molecule we just made in the previous step, is going to join into the cycle. What it's going to do, it's going to have someone, bring them into the club. You have to be acetyl-CoA to get in. Now what's going to happen is this molecule called oxaloacetate, it's a 4-carbon molecule, 1, 2, 3, 4. It's going to bind with acetyl-CoA. Remember, this now has 2 carbons. When that happens, coenzyme A is like, okay, I did my job, I got you in. It leaves. And then we now have a 6-carbon molecule called citrate. 1, 2, 3, 4, 5, 6. The little gray balls is what we're looking for. So 2 carbons plus 4, we're now back to a 6-carbon molecule called citrate. So the citric acid cycle kind of works like this. So the first part is we're going to take those carbons that we added, and we're going to basically just start cutting them off one by one. So we added 2. So here's our first one. We're going to kind of chop it off right there, and we got carbon dioxide released. And as we do that, we also pick up some electrons. And then we're going to chop off that next carbon. Boom, we're going to chop it off. Boom, got some electrons, release carbon dioxide. So eventually you get to this molecule right here. So we're back down to a 4-carbon molecule. And we need to start converting it back to oxaloacetate. So if you kind of think of this, it's kind of like 2 parts. The first part is to form citrate and then harvest the carbons that you put on. And then the second half is just converting this molecule right here back to oxaloacetate. So the carbons never really leave the cycle. They just kind of always stay in and just keep picking up the new acetyl-CoA. But in the process, we didn't really gain energy. We did get some little bit GTP right here, which is like ATP's cousin. Not much, but we picked up a lot of electrons throughout the process. So the last part, oxphos. So we're on the inner part of the mitochondrial membrane right now. So here's the outer, here's the inner. So what's going to happen now is we're going to take all those electrons that we've been harvesting through the previous steps, and we're going to kind of put them through like a little relay race. And what we're going to do is we're going to wind up generating a hydrogen ion gradient. So this gradient is going to drive this next molecule called AT synthase. So what's going to happen first is, remember, NADH, FADH2, so these are full taxicabs, full Ubers, full lifts. What they do is they come to these molecules embedded in the membrane, and they drop off the electrons. The electrons are then kind of like go to each of them, go through each little protein, and every time they go through, a hydrogen ion goes through. So every time an electron goes through, we get more and more hydrogen. So boom, 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 boom. We're going to accumulate a bunch of hydrogen right here. That's kind of like the first part. The second part is we have to use electrons to reduce oxygen to water. So oxygen is going to be the final electron acceptor So these electrons, after they go through all these proteins, they need to find a place to go. What they do is they hop on with oxygen, and we have water. So this is one of our products right here. Now all these hydrogens that we generated right here will then go to this next molecule called AV synthase. For every hydrogen that goes through, it causes this molecule to turn. And every time it turns, it takes an ADP molecule and a little free-floating phosphate, and it kind of just adds it back on there. So it's like, boom, recreate that bond. Now you have a fully charged ATP. So these link together. So remember, all the electrons we picked up in glycolysis and electron transfer, or in TCA cycle, we're going to pick them up, drop them off, go through here. Electrons get picked up by oxygen and make water in the end, and we generate this hydrogen ion gradient. This gradient then powers this molecule, and that will lead to the recharging of ATP. This is a cool little video that kind of walks you through how this molecule works. So bring it all back together, and I'm going to relate it to this equation down here. So the input is glucose. Here's your 6-carbon molecule right here. So remember, we went through, boom, 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 and we made a pyruvate. So we fed that into the TCA cycle. And basically, the TCA cycle was to harvest those carbons, and it got released as carbon dioxide. There's your 6CO2 right there. Now, eventually, we picked up a bunch of electrons. Those electrons were fed into the electron transport chain, where oxygen is sitting there waiting. And what oxygen is going to do is pick up those electrons and be converted into water. So here's one of your products, or an input right here that you did in glycolysis. You get a lot of product right here from the TCA cycle, your carbon dioxide. And then oxygen is an input right there, and we get it from picking up electrons to convert it to water. Now, ultimately, what you will see is you're getting ATP off this process. Most of your ATP will come from here. You'll get about 32 to 36 ATP off one glucose. So it's really more efficient to go through this process. Now, the entire process together is aerobic respiration. Now, if you go back to the very first slide, I kind of showed you how all the different pathways are linked. Depending on what the components are and how it's broken down, it might feed into different parts of the cycle. So this is kind of a little bit overview of that. So I will stop there. Like I said, there's going to be some review videos. I know this is kind of scary, but I'm going to put them with the fermentation side. So I'm going to stop it right there.